the do this. So this is jointly sponsored by the Johnson Comprehensive Cancer Center, the Biostatistics Share Resource, and the Genomics Share Resource. And today we have the pleasure of introducing Professor Dr. Pan Liu, and who is a postdoc researcher currently working with Professor Jessica Lee in her lab, the junction of statistics and biology lab at UCLA. And Dr. Liu joined UCLA in January 2024 after obtaining her PhD degree from the Department of Statistics and Data Science at the National University of Singapore. Her research interests include summary parametrics analysis, change point detection, subgroup identification, and also genomic analysis. She's currently working on developing statistical criteria to enhance the rigor in single-cell data processing. And so today, Dr. Liu will talk about gene set variation analysis, uncovering path-centric information from heterogeneous data. Pan, please.
Okay, thank you, Professor Lee, so much for the nice introduction. Hello, everyone. I'm really glad to be here to give this tutorial on gene set variation analysis. So, well, let's get started. This gene set variation analysis method, or GSVA, is actually an extension or a more enhanced version of the gene set enrichment analysis, GSEA. So today, we're going to start by briefly going through what is GSEA first, and then we will talk about why we need to go further to GSVA. And lastly, I will show you how to do GSVA in R using some practical examples. So the first question is, what is gene set enrichment analysis? Well, this is actually a method designed to identify whether a predefined set of genes shows concordant difference between two conditions. The two conditions can be, well, either healthy to control or to phenotypes. For example, in this plot, each gray dot represents a gene. We want to see these red dots, the gene set oxphus. We want to see whether this gene set would express differently between the two conditions, NGT and DM2. If we look at them from an individual gene perspective, actually there seems like there's no much differences because all of these red points line up pretty close to this solid black line. But actually, almost all of them are slightly enriched towards this NGT. So if we draw a distribution of their mean expression difference, this red distribution is actually obviously different from the one for all genes, which is the black distribution. And the GSEA method will be able to capture this kind of interesting pattern because it views this set of genes as a whole and compares it to the background, which is all the genes here. Well, that is the idea of GSEA. And it also somehow tells you how GSEA would work. So if we are interested in a gene set, like the red point in the previous slides, if we are interested in this gene set S, then we first rank the genes based on their correlation with these two phenotypes, A and B. We rank them. And then we want to see how the set of genes are distributed along this ranked list. Like this bar here tells you its distribution. And if it is like uniformly distributed along this ranked list, then that means there's no much enrichment. But if it assembles like to the top, for example here, assembling to the top, or assembling to the bottom, then that means the gene set is enriched in some way, either upregulated or downregulated. And we want to measure this kind of distribution skewness by a KS-like statistics, which is p-hit minus p-miss. These p-hit and p-miss are actually two random walks, or we say two running sums in some contexts. These are the formula for these two running sums. Well, we can see that this p-hit, it will increase whenever we meet a gene that is inside the gene set S. And on the other hand, this p-miss will increase whenever we meet a gene that is not inside this gene set S. And here the increment of p-hit would be this Rj, which is basically like the weight you assign to each gene. For example, you can give genes that are strongly related to the phenotypes higher weights, which means you place more emphasis on that. And then the difference between these two running sums will tell you how much difference is there between these two distributions. So it's like if this difference, we use ES to denote it. If this ES is large, that means that the distribution of gene set S is highly skewed among this ranked list, and we say it's enriched. And actually, this kind of task can be very powerful. However, one thing is that if we want to do the GSEA, we need to rank the genes first based on their association with the gene with the phenotypes. But what if the data set we are looking at is much more complicated? For example, what if we want to what if we are interested in like more complex or even hierarchical phenotypes? Then it will be very difficult to determine the ranks of the genes. And even if we can calculate the rank of the genes using some kind of correlation or association, we will also lose a lot of information if we just describe each gene set by a single enrichment score. For example, in the GSEA analysis, we just get a vector of GSEA scores, like one GSEA score for each gene set. So it is like the sample population information and phenotypic information are only used once when we calculate a rank, and then we just throw them all away when we do the further analysis. So in order to handle this kind of heterogeneous data set, an idea is that we can just do it in an unsupervised way. For example, I mean that we can ignore the phenotype information first, and then just derive a variation score matrix for each gene set corresponding to each sample. This means we will obtain a matrix instead of a vector. And then we can have the phenotypic information comes in later when we want to do all kind of downstream analysis. And this is like turning it into a start point, turning it from a start point, turning it from an end point to a start point in the analysis. And this is exactly the idea of gene set variation analysis. It will calculate the variation of each gene set within each sample compared to the sample population. Okay, this is the workflow for a gene set variation analysis. Actually, the pipeline is somehow similar to the one for the gene set enrichment analysis because it also ranks the genes first, like in the first two steps, and then it uses the KS statistics to calculate the variation scores. I think the main difference here is in the first step, which is like how we rank the genes, because this time we don't have the phenotype information anymore. We need to do the ranking in an unsupervised way. Well, the idea is that within each sample, like each column, we will need to rank the genes in terms of their relative expression level across all samples, like across the rows. So It's like the ranking is going to be within each column, but each gene's relative expression level, like the value we do the rank on, is going to be estimated within each row. That's the main difference between GSVA and GSEA. And to do this estimation, from a statistical perspective, we usually do a kernel estimation of cumulative distribution function, the CDF. This estimate will give the relative expression level of the gene I. This is the relative expression level of gene I within sample J. So, and if the data is continuous, like if it is a microarray data, then we will use a Gaussian kernel. If it is discrete, like a count data in the RNA sequencing, then we will use a Poisson kernel. Well, I think an example would help you better understand this idea. For example, let's say this is our input matrix, a gene by sample matrix. To estimate the relative expression level, let's look at the first row. Let's look at gene 1 first. We will line up its value along this x-axis. And the kernel estimation means that we kind of replace each single point mass. For example, it's 3.2. We replace this point mass with a kernel density. And we also do it for other values, like this Gaussian type density curve. And we think that, we assume that a true population will roughly distribute in this way along the several density functions. Then it is like we sum up all these densities to get the CDF curve, this curve from 0 to 1. And after we have the CDF curve, we can also calculate the CDF value for each point. For example, 3.2 would correspond to 0.1 on the curve. And that means we use 0.1 for the first gene, the first sample. We denote it by Zij, so Z11 would equal to 0.1. And we do the same for the second point, 3.9. 3.9 would correspond to 0.45 in the CDF on the CDF curve. So Z12 would be 0.45. And also for this for the third value, 4.7 is 0.88. We do the same thing for all the values in the first row. So here, this is the relative expression level for gene 1. And we repeat this process for each row. At last, we would obtain the CDF value matrix, or we say the relative expression value matrix for all the genes. And then based on these CDF values, we will be able to rank these genes within each sample. As we said before, this rank should be performed within each column. So we start to consider each sample or each column separately. We take one column at a time. What we do is that we rank the genes based on these CDF values. Gene with high relative expression will be put in the front. For example, the G8 here will be put at the first place. So this is the ranked list of the genes, and this is the ranked values from 9 down to 1. Well, actually, starting from this point, it is actually quite similar to what we have done for the GSEA method, because we just only need to deal with this ranked gene list. And these are actually the weights we want to use in our calculation. Remember, we also had weights for GSEA. Since we want to emphasize the genes both on the top and the bottom, we use their deviation from the middle value. So the weight is like in this shape. Okay, now actually we have everything ready to calculate the running sum or the KS statistics. And remember that p-hit and p-miss thing we did for GSEA, we also have this kind of running sums in GSVA. For example, this is the formula for p-hit, and this is the formula for p-miss. So back into our own example, suppose the first gene set of interest, GS1, includes 3 genes, gene 7, gene 3, and gene 9. And then this p-hit would be a black solid line, which increases whenever we meet a gene in this gene set. Here we increase at genes at G7, also at G3, and also at G9. Sorry, is there a question? Okay. And then this p-miss would increase at all other genes. This is quite similar to GSEA, but the only change here is that instead of looking at the maximum absolute difference, which is 0.76, this time we look at the difference between the largest positive and the largest negative. So we use 0.776 to minus 0.17. The reason for this is that the largest negative mainly comes from the members of gene sets staying on top of this rank list. And the largest negative actually mainly comes from the members staying lying at the bottom of this rank list. So we want to say, like, if this gene set has member both on the top and at the bottom, then we don't consider it as enriched. We want them kind of cancel out. So this is the enrichment score or variation score for the first gene set within sample 1 is 0.59. We would do the same calculation for other gene sets. If it is like this gene set 2, you can see that gene set 2 is like symmetrically distributed in this ranked list. If it is like that, then this variation score would be pretty close to 0. But if it is like this gene set 3, whose members just all lie on the top, then this variation score could be as high as 1. And this is for sample 1 or 1 column. We do that for all the columns one at a time. So eventually, what we get from the GSVA is also a matrix, but this time it's a gene set by sample matrix compared to the gene by sample matrix as an input. And this time the entry, each entry of this matrix would be the enrichment score of gene set J in sample K compared to the sample population. So from this output matrix, we can do all kinds of gene set-centric or pathway-centric analysis. Actually, just like the downstream analysis we did for the original input data, you can also do things like differential analysis, just that this time you can find differentially expressed gene sets instead of differentially expressed single genes. Okay, now we know the rationale of GSVA. Let's try it in R and see how it can help in our own analysis. Okay. All right, I created an R markdown file to present you with the code. Well, the good thing is that actually this GSVA method has already been wrapped up as an R package, and it is actually part of the bioconductor project, and it's very friendly to use. To install this package, we first we first need to install it using the bio manager. Since I have already installed it, I will just skip this step. And then we load this package. Well, to show you how this package works, I want to first I first want to illustrate how it would implement using a synthetic dataset. We would need a gene expression data matrix X, so generate it here. You see, this is a gene by sample matrix, where we have 10,000 genes and 30 samples. And we also need a collection of gene sets. And here we use a list to store it. This is how the list would look like. This is the name of each gene set, and here are the names of the single genes. We altogether have 100 gene sets, and their sizes will vary from 10 to 100. So these two are all we need. But before actually calculating the GSVA enrichment score, the GSVA package requires us to first wrap up all input as a parameter object, and we need to do it by calling this GSVA param function. So we put this expression matrix as the first argument and this gene set list object as the second argument. Then this function will give us a GSVA param object. We can feed this object into the main function of this package, which is the GSVA function, and it will help us calculate the enrichment score matrix. You can see this is the matrix. It's gene set by sample. We have 100 gene sets and 30 samples. So that is pretty much all about this package. It's really simple to use. But before we actually go into any real examples, I want to talk a little bit more about how to prepare for the input for this package. We will always need to call this GSVA param function to prepare an parameter object. But in the previous example, we only used the basic container. We used the matrix container for the gene expression data. Well, actually, we can also use some more advanced container, like the expression set object. Well, this object is actually provided in the bio-based package. And there's also another one called the summarized experiment object. The good point about using these two advanced containers is that you can also store the phenotype information and other experiment information inside, so that it would be more convenient for you to do the downstream analysis. And for the gene set, in the previous example, we only used the list, but actually you can also use the gene set collection object. By using this object, by using this container, the advantage is that you will be able to switch between different gene identifiers so that you can make sure your gene identifier for the gene set matches the one for the expression data. Well, let's start with the expression data. This is the basic matrix object we created earlier. It is like this. And we can just directly convert this X into an expression set object. We just need to use the expression set function and input X as the assay data argument. Then it will give you an expression set object. And as we said before, we can also store the phenotype information inside. But you just need to pay attention that you need to wrap up all the phenotype information in the annotated data frame object first before you can feed it into the expression set. So after you run this, you will have the expression set, with the X matrix as the assay data, and the phenotype information will be stored in the phenol data. We have two different variables here, the gender and score. Then we come to the gene set. This is the list object we just created. And you can also directly convert it into a gene set collection object by using this gene set collection function. But you will first need to convert each gene set into this capital gene set object. And you fit this list of gene set into this gene set collection function. Then it will give you a gene set collection object. And these are the names of the gene sets, and these are the names for the single genes. All right, so as we promised, we can feed in the expression set and gene set collection object into this GSVA function. The GSVA score we have will of course be the same, but the difference here is that the output will be in the format of an expression set instead of a matrix as before. Well, the reason for this is that the GSVA function will always give you an output in the same format as the input. So it is an expression set this time. Well, actually, in our own analysis, we seldom just generate these gene sets from scratch as we did before. The two of the most common way to get the gene sets are actually either from the gene ontology or from the MC database. So I will show you how to do that. First, if we want to obtain gene sets from a gene ontology term, then a very simple way to do this is to load the geoterm information from this human database in R. You first need to load this database, and then you can use the select function to get the geo information from this database. It's like you select the geoterm columns. And since we usually just care about the biological process geoterm, you can also further subset this data frame. So this is what you can get. It's a data frame with two columns. This column is the gene's name, and this column contains all the geoterm information of this gene. And in order to get a list object that we require as the input for GSEA, you will need to use the split function to group these genes in terms of their geoterms. And that is what we do here. After we do that, we will obtain a list of the gene sets. Also here, this is the gene set name, and these are the IDs of the genes. And this object can already be input into the GSVA function. But we can also convert this list into a gene set collection object by using this gene set collection function. Since this time we have the real data, we can actually specify the gene identifier that we use. We use the symbol identifier this time. Well, it takes some time to run. So you can see that here we have the gene set collection, and we have already specified the identifier type for this gene set. As we mentioned earlier, the good point about this is that we can easily switch between different gene identifiers, like using the map identifier function. Well, I tried it and I find that this function runs pretty slow, so I will just show you the result. After you run this map identifier function, it will give you another gene set collection object. You can see that this time the gene ID is the entries identifier instead of the symbol identifier, and the gene name is also changed from this to this. And another way of loading the gene sets is from the MSeq database. This is even more convenient to do. We have a get GMT function from our package GSEA base. Using this function, you can just specify the name of the GMT file you have, and you also need to tell it what kind of identifier you're using. And then you will be able to give you this gene set collection loaded into your R. And of course, to do this, you first need to obtain this GMT file. You will need to go to the MSeq database website. You can see that here we have different kind of gene sets for you to choose. In our analysis, I just chose the C2 gene set. And in the last column, you can just directly download the GMT files. These are GMT files for different gene identifiers. The one I downloaded is this one with the entries gene identifier. So you can see that here I have this file stored in my local folder so that I can just load it inside using the get GMT function. Well, actually, we also have some additional argument for the GSVA param function that includes the KCDF, max difference, ABS ranking, and tau. These parameters, they all correspond to certain steps of the GSVA pipeline that we just talked about. Well, let's show you that. Remember that when we were in the first step, we need to do the kernel estimation. And I mentioned that we need to choose from Gaussian kernel and Poisson kernel because Gaussian is for the continuous data and Poisson is for the count data. So you can specify which kernel you want to use by changing this KCDF argument in the GSVA function. And then this max diff argument is to specify how you want to obtain our GSVA score. If it is by default true, then it means the GSVA score is the difference between the largest positive and largest negative deviation. But if you set it to false, then the GSVA score will be something similar to the GSEA score. It will be the maximum deviation from 0. So it is just the red arrow. We don't minus this blue arrow. This ABS ranking argument is also about this step. If you set it to false, which is the default value, it will be just this one. But if you set it to true, then the GSVA score will be the sum of the largest positive and largest negative. So this just offers you certain kind of flexibility when you want to calculate your GSVA score. This tau parameter specifies the power of the weight in calculating the running sums. So in your own analysis, you can just choose different parameters based on your own prior knowledge or data feature. But actually, for most analysis, if you don't have much prior knowledge, then the default value would be suitable for most analysis. Right, so finally, let's come to some practical examples, some real data. The first data set we would like to work on is the glioblastoma multiform microarray data. A previous paper showed that This GBM has four subtypes that can be identified by different gene expression levels. So these four subtypes would be the phenotypes that we are interested in. And in this analysis, we just consider 4 gene sets. These 4 gene sets were proved to be signatures for some brain cell types. Their names are here. For example, the astrocytic up gene set contains the genes that are upregulated for this astrocyte cell type. Well, this dataset has already been built as part of the GSVA data package, so we can directly load them from this package. And this is the form of the input data. The GBM ESET data is the expression matrix, and it is also in the form of expression set object. You can see that we have the phenotype information here, the different subtype of it. And then the brain TXDB set is the list of gene set that we are interested in. We can view it. This is just the basic form of a list. We have the gene set names here and the single gene IDs here. Right, so what we do is just to feed these 2 input into this GSVA function. It will give us an output in the form of an expression set. And if you want to directly access the GSVA score matrix, you will need to use this exprs function that will extract the output matrix for you from the expression set. So here is the output matrix. We have a gene set. There are only 4 gene set by sample matrix of the GSVA scores. And then a very simple analysis we can do is to just draw the heat map of all the GSVA scores. Well, this is just the code to draw the heat map. You can view it on the right panel. This is a heat map of the GSVA scores for the 4 subtypes against the 4 gene sets. Actually, we can see that these 4 gene sets are pretty differentially expressed, and they would also be. Oh, sorry, I just noticed there is a question. It says, can we use matrix from Seurat object? Yes, of course we can. You can just first like in Seurat you can abstract the slot, the data slot or the count slot. You can extract that slot from the Seurat object, and then you will get a matrix. And after you have the matrix, you can just directly input it into the GSVA function. Okay, so if any of you have any question, you can just unmute yourself or type it in the chat. Well, so we can see that these 4 gene sets are pretty differentially expressed, and they can be identified as markers for the 4 subtypes in terms of the GSVA score. This is a very simple example, just for illustration purpose. And next, I would like to show you how to do differential expression analysis. Well, the data set we are going to look at this time is also microarray data. It has 37 samples, and of those, 20 of them have this acute lymphoblastic leukemia, and the other 17 of them have this mixed lineage leukemia gene translocation. So this is the phenotype we are interested in, or the subtype that is the ALL subtype versus the MLL subtype. So we can also load this expression matrix directly from the package, the GSVA data package. This data set is also in the format of an expression set, and we also have the phenotype data of phenotype information, which includes subtype. And this is the subtype information, 20 versus 17. Well, in this example, we do not assume that we have much prior knowledge about the gene set, and that is usually the case when we have our own analysis. So what we need to do is that we first obtain a broad collection of candidate gene sets, and then try to find the differentially expressed ones. Well, we can load this data. This object actually contains all the MSeq database C2 gene sets. And if you are interested in some specific subset, a sub-collection of the gene sets, you can also further subset this collection. For example, here we are only interested in the gene set signature of genetic and chemical perturbations. So we subset this collection in this way. And it will give us a smaller collection of gene sets. And then as we usually do, we can just feed in this expression matrix and this collection of gene sets into this GSVA param function to wrap up the input. Well, these two parameters here means that we want to filter out the gene sets that are too small or too large. So we only keep the gene sets with the size from 10 to 500. Then we run the main function, GSVA. Oh, well, this also takes a while to run because we have many gene sets this time. Okay, this is the output object, which is an expression set object. You can also extract the GSVA score matrix directly from it. So this is the gene set by sample matrix. And starting from this matrix, we'll be able to perform a differential expression analysis in a gene set-centric way or pathway-centric way. Well, in this analysis, we will use the lemma package to do the D analysis, but you can, of course, also use other package you are familiar with, like the DSeq or anything. But actually, the workflow for doing this is exactly the same as what we did in the D analysis for the original data. The logic behind lemma is that we first fit the linear regression model to the gene expression with the phenotype as the regressors, and then we want to test if the regression coefficients for these two phenotypes are significantly different. So here is how we do it. We first create a regressor matrix. And this is how the matrix will look like. It's a 0 to 1 matrix, and the second columns represent the difference between the two subtypes. So we are mainly interested in the coefficient of this second column. Then we fit a linear regression model using this lim_fit function. After that, we convert the linear regression statistics to a Bayes moderated statistics using the e-Bayes function. And And after that, we use this decide task function to perform the test with FDR threshold equal to 0.01. Well, let's see what this res will look like. This res object is actually a matrix of the test result. It's a gene set by phenotype matrix. For example, this 0 means that for this gene set, we do not reject the null hypothesis that the regression coefficient for this regressor is 0. So that means this gene type is not significant. Since there are just too many rows, too many gene sets, we need to use the summary to get our test results. Here we can see that this is a summary of the test result. We have altogether 21 plus 29 differentially expressed gene sets. And of those, 21 of them are downregulated and 29 of them are upregulated. As we usually did for DE analysis, we can draw a volcano plot. We can draw a volcano plot to show the result. So this is the volcano plot. We have the FDR threshold equal to 0.01. And in this part are the upregulated gene sets, and these red dots are downregulated gene sets. What we can also do is to draw the heat map of the gene set of the GSVA scores for only the differentially expressed gene sets or pathways. Right, so here is this is the heat map. We can see that these identified DE gene sets are indeed differentially expressed, and using these DE gene sets, we'll be able to cluster or identify these two phenotypes. Well, so that is all for this real example. One last thing I want to mention is that actually this GSVA function can also be run through an interactive interface. This is built together with Shiny. So what we can do is to run this iGSVA function. After we run it, an interactive interface will be pop up. So here in this box, you can input the expression data matrix either from file or from workspace. So here we just do it from the workspace. Let's just choose our synthetic one, the X matrix. And you can also input the gene sets. We also do it from the workspace. And this box enables you to change these kind of additional parameters, as we mentioned earlier. But here in this analysis, we just use the default values. Then we click on Run. It will help you to calculate the GSVA scores. I found that actually this interactive interface runs a bit slower than in RStudio, but it should be more like user-friendly. After you get a result, it will also show you the GSVA score distribution for different samples. Here, each curve represents a sample, and if you click here, you can also view the GSEA score, a GSVA score within each sample. Now we have the result. If we want to store it in a file, you can click on the download button. And if we want to save it back into the workspace, you can click the save and close. And after you click that, this GSVA score is already stored in this res object. We can see that the dimension of res would be 130, which is the gene set by sample. So this is the same as what we did in the R console. Okay, that is all for today's tutorial. Please let me know if you have any questions.
Okay, well, wonderful. Thank you so much for the very thorough and detailed tutorial. So I think we can accommodate a few questions.
Thank you for the talk. In the very beginning, I guess in general, can we input the quality of RNA-seq or microarray data for each of the cell? We're given like G1 for sample one, the quality of array, the measurement might be different.
Sorry, do you mean like instead of the gene expression, you want to input the quality, the sequence
Yeah.
the quality control? Is that what you mean?
Yeah, I mean, I guess the better question is first, like, can I input any kind of, like, let's say I ran like a GWAS across the genome, and then I p-value across the genome, and then I want to get, enriched set using that p-value, rather than microarray data or RNA-seq data.
Yeah, I guess that also makes sense. But
Okay, so
yeah, but actually I haven't seen that kind of analysis, but I think that would make sense because the p-value can also measure like the level that you want to calculate, like the association level, right? So you can also input that.
So I guess, okay. I guess I'm wondering, I'm wondering we can just put heritability across the genome and use that to obtain the gene set. Would that be sort of scalable in the paper, do you think?
Scalable in the paper?
I don't know if Han, the heritability. Like I'm just trying to see whether this can be applied in general, not just microarray or RNA-seq. I applied the p-value and report in different paper. I did like a epigenome-wide association study and then use a p-value to obtain the gene sets that are enriched for that like subtype, breast cancer subtype, or specific breast cancer subtype.
Yeah.
But I'm just trying to see like your experience. If I like use heritability that I quantify across the genome, feed that to the algorithm, and then get the gene set, would that be, from your point of view in gen, like whether it would be sensible?
Well, actually, I think from my perspective, it really makes sense. But I think that is really kind of an innovative idea to do the analysis, perhaps. But since I've already talked about the intuition of this GSVA, I think as long as the matrix you input is some kind of quantifying and have some quantifying scores, then it will make sense.
Okay.
Where is the matrix DK you can think?
No,
so
here
matrix.
here GSV is like a per sample, but in previous application, when I did with EWAS, I essentially got the gene enrichment for each of the so I'm thinking for one phenotype, I have a heritability across the genome, so I can get for that phenotype what is the enriched gene set given the heritability across the genome.
Yeah, I think that will be something like GSEA, where it's usually is the one vector and you just have the sets that you define.
Right.
So yeah, we could do that. And but for the heritability per se, I feel like the sets needs to be a little bit larger, not one snip at a time. So we may be able to do a gene at a time, but then depends on the number of SNPs that included in the gene, then that will be so yeah, that will yeah we could try this in general, like DK were saying, but then the sets to define heritability needs to be including relatively okay number of SNPs to begin with.
Well, if I had to impute it, like or
Yeah,
even 500,000, then gene set. I mean, gene set, was it like 1,000 on like the gene set size? Can
yeah,
it
if
like capture enough SNPs, right?
you have to, yeah, so just to define using imputed or something that to capture most of the genes and for the heritability. But then, yeah, I mean, it's doable. It's yeah, so it's something you have
Okay.
to. I was wondering for this kind of URNA GSVA, does the normalization impact the final result? Different type of normalization method will impact your result?
Yes, I think normalization has a great influence on the downstream results. Actually, you need to do the all the pre-processing, including the normalization, before you do this GSVA. And yeah,
So what kind of yeah, so you mean the normalization is part of a GSVA or
no, it's you need to do all of that pre-processing before the GSVA.
so yeah, so like there are so many different kinds of normalization method. What do you suggest that works the best with this package?
Well, to be honest, I haven't do that kind of thorough benchmarking for this kind of thing. But I think you can just choose the normalization based on your own data type. I think there have been plenty of normalization benchmarking papers in the literature, right?
It
Yeah.
won't so you mean it won't change the final result. Or like a differential pathways or something, you won't like no matter what kind of normalization method, the downstream pathway to declare to be enriched will be relatively consistent across different normalization methods.
Well, that is a very good question. I haven't tried that for that different normalization method. Yeah, but yeah, yeah, I'm sorry for that.
Yeah, great tool. You guys did a lot of like classer and yeah, it's very wonderful. Thank you.
Thank you so much.
All right, any more questions?
Hi, Dr. Leo. Nice talk. So I have a question like, if I need to transfer matrix from zero to object, is there any dedicated function for that, or how should I do that?
Sorry, can you repeat that?
So if I am using Surrey object, is there a way to transfer those matrix from Surrey object for GSVA?
You mean you want to transfer other types of objects into the matrix object?
Right. For single cell, people usually use Surrey. So those information is stored as Surrey object. So if I want to transfer those matrix.
All right, I thought I answered that question. You just asked that question in the chat box, right? Actually, I think for Surratt, there is a function like you can obtain the assay or slots of the Surratt object, right?
Mm-hmm.
So
no, I'm wondering whether GSV has something dedicated.
No, it cannot. It cannot directly work with the red object. You need to like extract the matrix first and then feed it into the GSEA.
Okay, got it. Thank you.
No problem.
Right. Any further question?
Just quick one. So is there like, have people done sensitivity analysis when you perturb the micro-seq data a little bit, and then you run analysis, gene set enrichment analysis? Does it change a lot?
Well, to be honest, I haven't done a kind of analysis before, so I'm actually from a more statistical background, so I don't have that many biological analysis experience as you guys do, actually.
Yeah, DK, just from my non-expert just perspective, I think you perturb your data just a little bit based on the way the scores are constructed. I do not feel that it might change the result very much. So
Yeah, I guess, yeah, I know it does like kernel. So, but I guess I'm trying to see whether I can incorporate that standard error, like of that heritability, like that uncertainty, like weigh it when you're trying to build the kernel. Maybe it's like sharper when it is higher quality around that one data point, like Pan showed, like each of the kernel has same distribution. But if a better quality can make it sharper or something, I guess what I was wondering if people already implemented it, so I can just use it as a black box.
Right, that is a very good thinking. But I don't think people have implemented that. You can definitely check the
Okay.
code instead of try to change the kernel.
Okay. Thank you very much.
All right, I guess we can stop here, and thank you, Dr. Liu, again for the wonderful tutorial for us. And thank you.